incredibly powerful toothcomb, which is characteristic of all the Strepsirrhines, but theirs is different in that it has grooves on the back that allow liquid to flow upward, that allow the venom injection, but it also allows incredible scraping of exudates from trees.
Hello, I have the pleasure of speaking with Professor Anna Nekaris, who is a professor at Angela Ruskin University in Cambridge. She's also the director of the Little Firefish Project and vice chair of the special section of the IUCN Primate Specialist Group for African and Asian prosimians. So welcome, Professor Nekaris. Could you please share a brief overview of your career and research?
I've been working on lorises since the 1990s. I actually did my PhD with a paleontologist, Tab Rasmussen, and it was his dream to have somebody study slender or slow lorises. I started studying slender lorises in South India. I moved on to working on slender lorises in Sri Lanka, where I named a new species. And after doing a lot of ecological field work, I got much more interested in conservation and seeing the horrendous conservation situation of slow lorises in Asia. I became the scientific director or scientific representative to help transfer slow lorises to Appendix 1 of CITES in 2007 and started long-term field project in Cambodia, working in Thailand, Singapore, etc., until I finally now have a very long-term project, which as of 2025, it's 15 years of job in slow lorises in Indonesia.
And what would you consider your greatest research achievement or the accomplishment that you're most proud of?
It's very difficult to say what is the greatest research achievement. There would be two sides. There would be more the behavioral evolutionary side and then the conservation side. From the behavioral side, it probably is the really exciting research I've been doing on the venom of the slow loris. Most people, when they think of primates or even mammals, they can't imagine that they're venomous. And even when I started working on slow lorises, when there was a suggestion that they were venomous, it wasn't even in my mind that I would look for that because most primatologists didn't believe it. And having done a long-term study on why they're venomous, what their venom does to each other, that it's used in interspecific competition, which is one of the rarest uses of venom in the animal kingdom, even among other venomous taxa like snakes and scorpions, etc. It's very, very fascinating. It's incredible. And it also has incredible conservation implications for things like translocations back to the wild and how they're kept in zoos.
What opinion do you hold that differs from the commonly accepted view in research on Strepsirrhine primates?
That's a good question. My greatest frustration when thinking about nocturnal primates in general is this archaic, ridiculous, out-of-date view that they're solitary. And so the more and more people who study nocturnal strepsirrhines with technology, including radio tracking, accelerometers, night vision cameras, we see that many of them, if not most of them, are certainly social to some extent. And in the case of the slow lorises, they live in small family groups. They're incredibly social. They play with each other. They groom each other. They have long-term romantic, deep, affectionate attachment to each other. And it's so interesting in terms of primate evolution. It's interesting in terms of evolution of social organization. And it just takes patience, time, and now in these days, this fantastic technology that undoubtedly shows this really deep sociality. And it's one day going to be my mission to get rid of this term solitary for primates.
And is there differences among different lorises in terms of their social behavior and social interactions?
So within the different species, where slow lorises are still so little known and lorises in general, but even back in my first studies of slender lorises, it was obvious how social they were. And it wasn't a long-term study, and we never were allowed to do genetic analysis. So we knew that these groups were sleeping together, and we assumed, like a lot of people would have thought, that it was maybe a male with multiple females. But now having long-term data where we know you have a male, female, and 1 to 4 offspring, I think even those earlier studies probably could be reevaluated now as to what was happening in those groups. And even for some of the bush babies, where it was assumed that it was groups of females, this might not be the case because the genetics, etc., weren't done at the time. It was just assuming that was probably a harem social organization would be something we would see as an ancestral social organization. But when we look at reconstructions of various well-studied nocturnal primates, monogamy keeps coming up as the rule, or unimale, unifemale social organization, and it does differ. You have animals trying to get together and they reject each other. You have others that kind of fall in love at first sight. You have others who, we had a female who her male was killed, and it took 2 years for her to find somebody else to hook up with. So she lost 2 years of reproduction because she couldn't find a replacement for that male who died. So yes, I think as we get longer term studies of the other species, we'll start to see other variations as well.
Thank you. And could you provide a brief overview of the recent evolutionary history of Lorisiform primates?
A bit difficult question for me, and I've looked into this. I mean, one of my very first papers was a overview of Lorisiform evolution with my PhD supervisor, Tab Rasmussen. And to be honest, not a significant amount has changed since 1997 when we published that paper, because the fossil record for the Lorisiforms remains scanty. We don't really necessarily know which was the fossil Lorisiform. We have some from the Miocene in Kenya. We have some examples from Egypt where we might have potential Loris Galago ancestors. But I think what we do know is with fossils of Myoeuodicus from Kenya, which is a very interesting group, and it wasn't sure if it was more of a bush baby or a loris-like primate, is with new and amazing studies of their inner ear, where we could look at how the ear might be predictive of locomotion. Myoeuodicus seems to have been a slow climber, which is really exciting that we have this slow climbing, really crazy, weird, unusual adaptation for the primates happening in the Miocene. So we're still searching for additional fossil ancestors in Asia, but it's the fossil record is so scanty because of so I think this is going to be one of the great chances for a student to find fossil deposits in parts of Asia that can yield lorises. And the suggestion is that maybe they are not so speciose as other primates today, and maybe they were also not so speciose in the past either.
What are some of the key challenges involved in conducting research on nocturnal primates, and how has your approach to researching these different groups evolved over the course of your career?
It's always been a challenge to find a field site to study slow and slender lorises. One of the key challenges is, unlike jumpy, springy bush babies and tarsiers, lorises won't enter traps, or will very reluctantly enter traps. Or if they do enter a trap, they have a very long body, so they could have a limb on a branch and they could snatch the food from the trap without getting stuck inside the trap. This means we can't radio track them and radio collar them or recolor them. And that if we want to catch them, we need to climb a tree with a glove and pick them up. So in the more rainforested habitats where the trees are very, very tall, and connected, it's much harder to catch them in that sense. And it's also much harder to watch them because the forest is so dense. So for all nocturnal primates, visually seeing them at night is a huge challenge. And again, some technology, let's just say infrared torches or heat cameras, can allow us a little bit better to see what's going on. But in the really dense canopy, it doesn't help that much. And yeah, so that's always going to be a challenge, is just the catching, watching, observing, of course. But another major problem we face that, for example, the researchers in Madagascar working on lemurs don't face, is that we have lions and tigers and bears and elephants. And these are all very, very dangerous animals at night. And you would be amazed how quiet an elephant can be and how you can bump into an elephant's leg at night, which has happened before, and be really terrified. And it's very challenging to run away as well. So many people won't let you study nocturnal animals at their field sites because of the fear of these larger animals. Many trackers or like field assistants won't go out with you at night. And beyond all of the real dangers, of course, the night is a time that many cultures in the world see as something spooky and scary. And the number of places I've been where people refuse to enter the forest at night for spiritual and ghostly reasons is a true barrier to research in some places.
Do you think this could be one of the reasons there is less research on these primates compared with other primate groups, or do you think there's other things going on?
We could even call it a Discovery Channel syndrome. A lot of natural history films over and over focus on the same group of small charismatic species. And the same with zoos. You go to zoo after zoo, and the smaller, lesser known species may be less featured, maybe in a smaller exhibit, maybe in the corner of the zoo. And so I think there's a lot of marketing in the way students learn that they don't even, it's not even on their radar that they could study these species. And it's not just within the primates. If we look within all the other major groups, say, of mammals, and we think of the cats, we don't have the small cats being studied. Or if we think of the wild dogs, we don't have the smaller dogs being studied as much, especially in the tropics. So I think there's just this marketing that the really big, amazing, charismatic animals are more important or more interesting, then funders want to continue to fund those. And then the way research works, if you study chimpanzees, your first paper can easily get 50 citations because so many other people are studying chimpanzees. But a slow loris paper may take 10 years to get 50 citations. So the way that research works, it's not, it's against you to study something new sometimes. But I always tell students that there are so many amazing species, many of them smaller, be it some of the smaller monkeys from South America, some of the really challenging rainforest colobines in Africa, or almost all of the nocturnal primates are ecological niches for students that they could jump in and be the world expert in 6 months if they would just take the risk and study that species.
Thank you. You've already touched on this earlier, but could you summarize what we know about slow loris venom, including its ecological and evolutionary significance?
A few theories as to the why slow lorises might have evolved venom, but first, what is it? They have a two-step venom system. So like most prosimians, they have scent marking glands, and the one that is very prominent on the slow loris is the brachial gland, which is not in the armpit and is not in the elbow, as you read online very often. It's just in the upper arm. And it very neatly, when they put their arms over their head, is almost exactly level with their mouth. And they could turn their head from side to side and lick the brachial gland. And it combines with saliva in their mouth. Both the brachial oil and the saliva contain a number of unique volatile compounds. But and in fact, on its own, the saliva can cause intense pain in laboratory experiments, and the lethal effects come when they are combined together. So if we do in vivo experiments with mice or with various insects where we've applied the venom to them, the brachial oil causes a reaction with the saliva that that essentially creates a protein reaction that creates the venom. And this is a cytotoxin because it can cause extreme necrosis. And in slow lorises, when they bite each other, the necrosis can be so horrific that it can eat through their skull or eat into their brain, and it frequently causes death. It frequently causes death in transport and the illegal wildlife trade because lorises are crammed in cages together, bite each other. And so we found very much that at least now it's used for interspecific competition. But why would lorises need interspecific competition? And it's not so common, like I said, in other animals. The platypus is another venomous mammal that uses venom for interspecific competition. What we think potentially is that when lorises were moving into the Miocene during this period of global cooling and having to move to the ground as a slow climbing animal, which we already know from this amazing set of fossils in East Africa, that we had slow climbing animals before they moved into Asia. It's really risky because when you're slow climbing on the ground, you can get eaten very, very much. So the other genus that was moving into Asia at the same time is Naya, the cobras. And slow lorises have extra vertebra in their spine, which is also weird. They have a stripe down their back that changes seasonally in several species. They have the stripe moves onto their head to form this crown with these spectacles around their eyes. And when they are threatened, they either hang from their hind limbs from a branch and sway, or when they also can stand on their hind limbs and sway, and they look incredibly like a cobra. People who have kept cobras say the loris hiss is very similar to the cobra's hiss. And there have been people who have mistaken their pet loris for a cobra. when they've come into the room and seen it swaying spine, for example, attacking a house cat or another intruder. So it's very interesting to have that suite of characteristics, including the fact that their saliva with this brachial oil is toxic. So we call this Mullerian mimicry, where potentially moving into an environment with an actual venomous animal that also slithers, that has this spectacled features on its hood when it stands upright, would have allowed lorises to exploit that niche of mimicking cobras, including with this venomous bite. What was also happening at the same time that's incredibly interesting is this move to extreme exudativory. So lorises are the most exudativorous primates. They can eat gums that can give a human third-degree burns, and they have very large granules in their saliva that seem to allow them to put this very toxic material into their mouth without themselves getting burned. They lack whiskers, so they're also lacking obstruction on their face that allow them sort of to burrow in and eat this gum. They have incredibly powerful Toothcomb, which is characteristic of all the Strepsirrhines, but theirs is different in that it has grooves on the back that allow liquid to flow upward, that allow the venom injection, but it also allows incredible scraping of exudates from trees and gouging, active gouging. Slow lorises begin to actively gouge by 4 to 6 weeks old, so they're already gouging when they're babies. And by the time they're older, they get this incredible array of features on their teeth that I'm really hoping one day someone finds in the fossil record. It looks like little tree rings. They're kind of like Harris lines of showing when they're developing, and when they're gouging very, very hard, they get these sort of, yeah, I don't know, you call them lines of I can't think, stress from gouging. So really incredible, interesting animals in terms of gouging, but potentially the change in the saliva that led to the ability to digest this incredibly toxic gum could have been in that cocktail of evolutionary complexity that was happening during that period that led to their becoming venomous. We don't have evidence that they use it against predators. Their predation strategies are really cryptic, and we don't really have evidence. There's potential evidence it's useful as an anti-parasitic element when they lick their fur. There was a BBC film years ago that filmed a slow loris mother licking its baby at the Duke Lemur Center. And it got into the literature, slow lorises lick their baby with their venom. It's not true. We have no evidence of that whatsoever. So in this video, I'll state that yet again. I've written in a few papers, but the BBC wins all the time with this single observation in 1992 of a film man at Duke. It's kind of, so no licking babies with toxic poison. I mean, maybe they do, but it's not the main function of it. And they can actually overlick their babies and their babies can die.
Thank you. You mentioned the Strepsirrhine toothcomb. Just wondering, is the idea that it evolved originally for grooming and then was perhaps further utilized for gum eating, or is this something where there's ongoing research into the origins of this particular feature?
A very interesting question, because we don't have really good data. There's not really any toothcomb primate in the fossil record. We have Caranacea, where there's really good evidence that it might have done some scraping of gum. There's been some suggestions of some of these putative Lorisiforms that there's lines on the teeth that suggest they abrased hair, you know, from the fossil record. But if you think about it, in fact, what would be a really good selective pressure for toothcomb? Why would it be grooming? It seems to me much more sensible that it would be exudativory. So I think that's going to be another great discovery if anyone ever finds the first proper full toothcomb in the fossil record, and looking at the environment in which it was found. And because I feel that exudativory nowadays is not so common, but if we look at the other main crazy, weird exudativorous mammal, it's a colugo, which is sort of a living Adapiform in some ways. So some people argue that colugos at least come from Adapiforms. And yeah, and that they're really weird. They look like a, you know, dinosaur, alien, strange mammal that should be starring in a colugo version of The Ring, Japanese horror film. And they have those strangest little toothcombs that are for gouging gum. And someone suggested once their toothcomb was for eating leaves, but now we have more and more studies of colugos, and they're also fantastic gougers.
you. And the next round is a quick fire round. So I'll ask the one on the fossils first. So do you think the common ancestor of Lorisidia was an Adapiform?
I would say with the current information we have, it is the most parsimonious answer. So it's very unlikely to be an Omamiad, for example. But I think that there are some good generalist examples in the Adapiforms that could have led to a Loris clade along the way, and also some of the other living Strepsirrhines. It's also with the biogeographic distribution of Adapiforms makes sense for that. And I don't think we can only say it's the most parsimonious answer at the moment. We can't be completely definitive because there's an ancestor out there to be found that would answer that question more definitively.
Does the cryptic nature of many Lorisidia species help or hinder conservation efforts?
I suppose you could say it hinders it in a couple of ways. The first way is it can be hard to see them. It can be hard to survey them. The potters and anguantibos are particularly notorious. I've had so many students try to go out to start a study of anguantibos, which are the least studied of all the primates, I would argue. And I mean, it's the least studied primate genus, at least. And they're so difficult to find and so difficult to see. And they do sometimes curl up in a ball and hide their face. So at night, you're relying on the tapedum lucidum, that shining eye with a red torch to find these animals. And yeah, the anguantibos are pretty horrible. If we think also about how crypsis can affect their conservation, they're brown animals that they have their own species mate recognition systems that aren't the same as the diurnal primates, so they kind of look the same. And there are problems like translocations of different species into the wrong country, the wrong place, because many people don't want to accept the splitting of the nocturnal primates into multiple species. I think in the case of Madagascar, where we have so many lemurs, it's frustrated people. So, but again, if you take that example of the Bengal slow loris from India to China, where in the same geographic range, there are so many gibbons, so many macaques, so many langurs, and one loris, it seems to me not even scientifically logical to have one slow loris. And so, but that cryptic nature, again, makes it really hard to catch them, really hard to get genetic samples. And the regions where they live, it's really hard to do genetic analysis just because of legal reasons as well. So one day, again, we will know more.
I remember reading some of your research on, I think it was loris groups getting released into the wrong habitat. And I wonder if you could talk about kind of the how common this is and the main issues that it has for primates in the wild.
Yeah, I mean, this is a typical invasive species problem. In the UK, we have it with red and gray squirrels, and almost any person in a supermarket would know that it's a problem. So I find it phenomenal that educated people who are doing releases just say, oh, I think they look the same, so I'll just release them. And I've come across completely the wrong. The pygmy loris recently was named as a unique genus, and there was a Spanish television show, natural history show, where the presenter released 100 of them into a forest in Thailand as a goodwill gesture. And that it's never even existed. It's a genus that never even existed in that country. So these are the kind of things the public like to see as well. They love the idea of release. Students love it. They love the idea of wanting to work in a rescue center. And I think they need to really reevaluate the importance of keeping these animals in the wild and other conservation actions that don't require them to be in rescue centers, that then don't lead to all these additional problems like invasive species. I feel that for certain species, it could be a really big problem of releasing them into the wrong place, especially on small islands. And it also could cause problems to ecosystems where prey are available there that have never seen that kind of predator before. So it's not only the loris or the macaque or whatever animal is being released in these places, it's all the potential things they may prey on that could have dramatic effects as well.
More of a lighthearted question. Do you have a favorite loris species? And if so, what makes it stand out to you?
I have to say the Javan slow loris, because it's the one I've been studying for 15 years. They are just so beautiful. They have the most beautiful face mask. They have the most wonderful ears, the most beautiful different colored babies that change color as they get older. And I just know so much more about them. I would say on the opposite, if I had the dream species, it would be to study, it would be the golden anguantibo, the Arcticebus calibarensis, sorry, Arcticebus aureus. It's very, very, very beautiful as well. And we know so little about it. It's would be like the golden animal to study, to know more about. But for the one I know, so it's the one I know the least about and the one I know the most about.
Just you mentioned the face mask. I wonder if you could go into some more detail on this and how you conduct this research.
Okay, so for different species, most of that work was done in museums, and we visited several museum collections, and I use a camera with the same exact flash and same exact background, and I use Munsell color charts, which are standardized soil colors, and you're able to then, when you put your photos into an online system, you could standardize the colors using red, green, blue codes. It's really interesting because colors are sort of circular, so they're really unusual data to have to deal with. So we had to find statistical tests that would accommodate this strange distribution of data, and there are R packages now that allow for analysis of RGB colors, so we were able to do that as well. And in fact, we also used colorblind simulators because lorises don't see in color, they're monochromatic. So we were interested how lorises see each other, how their predators may see them, and how, let's say, dichromatic predators and trichromatic predators may see them. And in fact, we found the most in the single population, in our single population in Java, we found more differences in the contrast of the black and white, suggesting that the face mask is used within the species for communication and identification. But also with the museum work, we found that there are patterns of face mask difference that probably are uniquely evolved characteristics. So for example, in some species, they have completely naked ears. In other species, they always have a face mask that extends to the jawbone. So the face mask is very long, and there's another species with almost no patch around the eye that just has a little black ring, and it's consistent in all individuals in that species. So using those as character states, we were able to use discriminant analysis to show that they fit into different species. And subsequently, we've done genetic work, and the genetic work by and large follows the patterns of the face masks, with a little bit of variation in Borneo, where there's probably even more species.
So the final question is always outside of your research directly. So you played a key role in public outreach in shaping how lorises are perceived by wider audiences. So could you describe some of the main threats these species face and how increasing awareness, public awareness, can make a difference, and the role of initiatives like the Little Fireface Project in addressing these challenges?
Okay, so in 2011, I was invited to make a film by the BBC. And this came up after slow lorises were moved to CITES Appendix 1 because of this dramatic trade in slow lorises. They're traded extensively for traditional medicines. They have over 100 uses in countries like Myanmar, Cambodia, and Vietnam, for example. They are traded as photo props on beaches where tourists can take photographs with them, with a beer or whatever. They are widely traded in the pet trade. And the pet trade really came to light in 2009 when a single video of a slow loris being tickled in Russia in a flat became viral. It went viral. It was supported and shared by many celebrities. And it's now become almost completely decontextualized. You could see it as a meme. You could see it on coffee mugs. It's actually even become synonymous with King Julian from Madagascar, which has nothing to do with the slow loris, but the public doesn't distinguish that. And essentially, more people knew what a loris looked like in a pet situation than in the wild. And the first time they might have ever seen one was as a pet. So the BBC wanted to capture this in a film, and we actually had a really stark scene of lorises in illegal wildlife trade in Indonesia, and they said the public will want to know what to do to help after they see this film. So I started my charity, which is the Little Fireface Project. Little Fireface is the Sundanese word for slow loris. The slow loris has wonderful names throughout their range: the shy, the wind monkey, the ghost monkey, the moon face. But I really liked the little fire face because of the patches around the eye. And I always wanted my project also to help other nocturnal animals with those wonderful face masks as well. And yeah, so in addition to, so as part of that project, we launched massive social media campaigns. We did additional films, including in Loris range countries on national television, did a TED talk about the topic. So a number of all the social media we've been doing. And even I was in the airport the other day wearing this slow loris necklace, and a lady said, "Hey, that's that cute animal we're not allowed to have as a pet. Isn't it poisonous?" I said, "Actually, it's venomous." But I thought, that's my research. That's amazing. The lady at the airport identified it just from a necklace. It's really incredible to see that happening. And so I think it's something really important when we're studying. Everyone who studies an animal in the wild now has a role to play in its conservation, because the world is changing so fast, habitats are disappearing so quickly, that if we are only concerned with as interesting as all of the evolutionary questions are, and I love them, and they were how I started my career, we can't answer any of those if those animals are extinct in the wilds. And if they're extinct because of a ridiculous reason, like people wanting to make a nice TikTok video, it's even more shameful and sad. So if we could use videos like the one you're making today to send out these messages to a younger audience who has that passion to carry on these fights and to help save the environment, that's a really great thing to do.
Thank you. And I'll add a link to the Little Fireflies project below this video if anyone wants to learn more or find out ways they can help. So thank you so much for doing this interview. It's been a pleasure speaking with you.
Thank you very much.